As I said, thanks to the organizers, today I'll talk about the activity which was recently published on PCDH19 by using the IPS model, which was exhaustively dealt with by Jack Parent. And after that beautiful introduction made by Jack Parent, I only want to underline three aspects which are very important to understand the IPS importance, i.e. the fact that they are used in neurological diseases that are until now were so very hard to study on man because the tissue is highly sensitive and delicate, the human tissue, I mean, and it can't be analyzed in a detailed manner, which is the case with other tissues where you can have biopsies, whilst IPS, once taken from the skin of the patients, allow you to reprogram fibroblast and pluripotent cells and then differentiate neurons. They are also important because in some cases the murine models and the mice models tend to capitalize perfectly the model of the pathology. Another important thing is that they can be utilized in the form of tests for pharmacological screenings that allow to observe the rescue of the phenotype and the recovery of the patient's phenotype, and at the same time to test the drug on other cell types, cardiomyocytes, or any other myocytes that may be then differentiated. And then we could also try and understand what is their future application in regenerative medicine, although we're not ready yet for this system. I will now show you the IPS colony, the way they appear to us, and how we can differentiate them in different neuronal types. Starting from time 0, here we have a cell colony which is clustered together. And after the in vitro manipulation with different protocols, you can see they could originate cerebellar neurons, motor neurons, and cortical neurons that are recognizable according to specific markers that were developed for these neuronal types. Another IPS advantage is that it allows us to study the cell before and after the differentiation, but also during the same differentiation. Here we have the simple microscopic pictures that, starting from time 0 and day 0, show you the progression of cells during differentiation, how they get reorganized, because starting from the colony after some 10 days of differentiation, they take up the typical neural rosette shape. And after that, they keep growing in the next 5 days. Then they become disorganized in individual cells, that is, future neurons and neurites. And then later on, they will extend and form synaptic connections. And here we see the positivity for neuronal markers. This is the beta-3 tubulin in this case. The reasons why it is important to study the role of the PCDH19 in this cellular model has already emerged from the previous presentations and contributions, but here we have three main reasons listed. One is the possibility of investigating the neuronal target type of the condition, that is human cortical neurons, then the possibility of studying the cellular and neuronal characteristics during differentiation, and naturally also the possibility of observing the characteristics of the cell and of these neurons in cells that were taken from patients. I wanted to underline three aspects that are fundamental at the time of the development of brain. One is the formation of the neural plate. This is the neuroectoderm, and which will then generate the brain. And this is a highly complex structure, but one of the very first phases of the development of, at the time of the neurogenesis, is this sheet, which you can see here as a tiny microscopic slice. This is the sheet, which then becomes curved and originates the neuronal tube.
Successivamente
And the cells that are then expressed internally to the neural lumen will then give rise to neuronal progenitors, or they may generate neurons. And they will migrate outwards, thus generating the different layers of the cortex. And this is what they generate. Camillo Golgi already in 2075 had tried to understand, and this is the layer of the neurohectoderm, but this is a structure which is not yet clearly understood. Our effort is the one of reproducing in a simplified system, i.e. in vitro, starting from the IPS, the different stages. And what we know until now about the in vivo role of the PCDH19 is something that we have derived from the zebrafish model. Maria also showed something more about the mouse model, which allows us to understand this function. And the zebrafish, with this respect, we can observe that the PCDH19 has a role in the morphogenetic movements, because here we see a situation which is a typical controlled situation where the protein is working and the cells are orderly migrating and organized, whilst when the protein is reduced or abolished, the movement of cells during the formation of brain, that is during the neural tube and neural plate formation, is highly disorganized. And the cells here have also a morphological alteration which prevents the self-avoidance, which means that the dendrites will not overlap. And this is avoiding the redundancy of synaptic connections. So PCDH19 is playing an important role in the morphogenetic movements of cells and in the morphological organization of the neuron itself. We tried to understand how PCDH19 was expressed in human cells, starting from the IPS at day 0, that is before the differentiation. And what we observed is that this is the IPS colony, and the localization of the protein in most cases is located only and localized at a single cellular pole. And equally interesting is that we see a two-pole localization in cases where the cells are undergoing mitosis. This localization was observed for a different protein already, which is important for the development of the nervous system, which is the ISPM. And in this case, the protein is a cause of microcephaly in man. It regulates the symmetrical cleavage of neural progenitors, and I will later show you the pictures. At the time of the differentiation, the localization of protein is more organized, whilst the APS was rather random, and in each single cell there was a focal expression point, but at neural cells, you can see larger ones and smaller ones. The PCDH19, marked in green, is localized at the center, together with a marker of the lumen of the neural rosettes, which is ZO1. The important aspect of this localization is that at the stage of neural rosette is very much comparable to what happens in vivo within the neural tube. And just like in the neural tube at the central level, where the lumen is, where the mitotic red-colored cells are, which then proliferate, and we see that the differentiated ones migrate towards the external parts. And here in the rosette, we've noticed that this is what happens inside the rosette, while the proliferating cells are at the center, but then once they differentiate, they migrate towards the external part. So this is easy to correlate in vitro and in vivo. We are making a very quick parenthesis as to the symmetries and asymmetries of the neural progenitors, because it's important to establish the differentiation of the cells themselves, because if the cell is dividing in a symmetrical way with respect to the base of the neural tube or of the neural rosette, then the two cells, daughter cells, will be neural progenitor cells and will strengthen and reinforce the pool of cells that proliferate. But if the division is asymmetrical, then one cell will be staying closer to the lumen of the tube and will be the neural progenitor, whilst the farthest cell, which is farther from the apical projection of the tube of the rosette, will then differentiate itself by migrating outwardly and originating neurons. Taking a closer look to the rosette, we've also seen that there are two cells, especially two cells that are then dividing. One is the A cell, which is dividing itself in a symmetrical way, especially with respect to the base and the lumen of the rosette, whilst the other one is doing it asymmetrically. So probably these cells will still stay progenitor cells, but proliferating progenitors, whilst here this one will be a progenitor, and only when differentiating itself it will migrate outwards. The important thing is that the PCDH19 once again has been localized in such a way as to orient the division plan of the cell. In one case it's perpendicular to the lumen, and in the other case it's running parallel to the lumen. Here we see a video showing you how to best visualize the neural rosette, the PCDH19 localization marked in green inside the lumen, but at the same time at the two poles of the cells, which are then been split, you can see them marked in yellow here. And then, quite naturally, the important thing is that the localization of PCDH19 is important at the level of differentiated neurons. And the localization is localized at the pole of a cell. And here we see it in the contact points where the cellular body is in contact with the cellular body of another neuron, and also between the cellular body of a neuron and the terminal part of a neurite of a different neuron. In conclusion, out of this results, we could see that these are the controls obtained from healthy subjects, and we can say that the PCDH19 may play a role in allowing the cell to understand in which position it finds itself, especially with respect to other cells. And this happens in cells that are proliferating, such as the IPS colonies at 0 day, and also those cells that are heading towards the neuronal differentiation. And we could also say that PCDH19 may be involved. This is visible on the zebrafish studies, but also in the case of specific localizations at the central portion of the neural rosette may be involved in the patterning events which allow for the development of the nervous system, and especially of the neural tube in man. In future times, it is going to be important to understand what is the phenotype of the cortical neurons and how the rosettes behave within cells that were harvested from patients that are having the condition, that have the pathology. Having said this, I'd like to thank Dr. Arrigo Bertini Dr. Alessandra Terracciano, sorry, and all of those who have given a contribution to this activity, the clinicians Marina Trizano, Nicola Specchio, the collaborators Curaci and Di Roche from Japan, Stefania Orini for the microscopy activity, and all of the others working just like I do in the differentiation field, the IPS differentiation field, and most of all, the sponsors and BNL. Thank you.
You didn't show the validation of the antibody. Can you tell me that it really detects PCDH19? Yes. So have you done shRNA downregulation of PCDH19 in these cells? And did you lose all that? Yes. And so that was our only question, to see that really you're picking up PCDH19, because we all know that there were problems with antibodies against PCDH19.
Claudia, parla dentro il microfono.
Yes, but also we tested it with after silencing the gene, and we saw a reduction. So we can be fairly sure that it is the right that it has the right specificity.
Jack?
Claudia, did you see any phenotype when you silenced the gene?
So actually we did it also at T0. We did the silencing and then we just extracted. We are pursuing with the experiments that are ongoing, so